In just a moment, we have Dr. Donald Vinn presenting Translating Microbiology of Infections into Genetics of Immunology. We've got 45 minutes allotted for this session, and we're going to stick to time. If time permits, we will take questions at the end of the presentation. Disclaimers. Please note that the information presented during this session is not medical advice, nor is it intended to be a substitute for medical advice, diagnosis, or treatment. Always seek the advice of your physician or another qualified health provider with questions concerning a medical condition. The Immune Deficiency Foundation education events offer a wide array of educational presentations, including presentations developed by healthcare and life management professionals invited to serve as presenters. The views and opinions expressed by guest speakers do not necessarily reflect the views and opinions of the Foundation. With that, I'm going to briefly introduce our speaker. You can find his full bio in the app if you are interested in looking at that. Dr. Vinn is an infectious disease physician, medical microbiologist, and section head at the McGill University Health Center in Montreal, Canada. As the director of the Center of Excellence for Genetic Research in Infection and Immunity, his translational research program aims to decipher how molecular problems of human immunity contribute to infections that are recurrent, recalcitrant, and/or unusually severe in patients. Welcome, Dr. Vinn, and thank you for joining us.
Well, great. Thank you very much, Catherine, for that introduction. I'd like to thank the IDF for inviting me all the way up from Canada to talk to you about this topic, which holds dear to my heart. As mentioned, I'm an infectious disease physician. I'm also a medical microbiologist, which means that in the diagnostic microbiology lab where people get their infections diagnosed, that's where I work. And I also do research in people who have inborn errors of immunity. This title is a bit misleading because it seems all formal and scientific. How do we translate microbiology of infections into genetics, into immunology? I think the real title of this talk is Where the Rubber of Immunology Hits the Road of Life. And I think you'll understand why as we sort of go through the different components of this talk. So as an infectious disease physician, our roles are very simple. A person comes in with symptoms, we try to determine if it's an infection or not. If it's an infection, we try to figure out what organ system is infected, we try to determine what bugs are causing that infection, and then we try to select antimicrobial therapy based on either the bugs that are identified or the organ that's infected. And we all know that they come with different types of syndromes, and we have a very syndromic approach. You come in, you have infection, we find it, we treat it. And that works well for the majority of cases because, in fact, most physicians can treat infections. But of course, there are some infections that are unusually severe, and we'll get into some of those examples, or just unusual, like your physician can't pronounce the bug that was identified, right? Or it's recurrent. It's like, wait a minute, this is like your 17th pneumonia in the last 4 months. And so when there's a problem there at the level of the infection, that's where we get involved as an infectious disease consultant, and that's where I particularly get involved if there's no obvious reason for why this is happening. Because at the end of the day, when you have an infection that can't be adequately treated, you have to go back to basic first principles, right? What is an infectious disease? If you can imagine the blue line of cells, well, that's our body's barrier to the outside world. And the outside world is nasty. It's full of germs out there, right? And those germs, some of them can't infect humans, some of them may infect humans, some of them want to infect humans. But we're not just a layer of blue cells, right? We also have our own army that is intended to be mobilized at the right time to try to fight the bugs. And at the end of the day, an infectious disease is really the result of cellular warfare between the microbiology and the macrobiology, or the host army's response. So we're going to go through all of this. And I apologize ahead of time, but to understand this talk, we're going to have to talk about 3 different components: immunology, genetics, and microbiology. So this is, from the far side, the room of hell. The 3 rooms actually converge into the same thing at the end, and you'll see that that room is called inborn errors of immunity. So first off, immunology. I don't need to explain to most people here what immunology is, but we'll make sure we're all on the same page. Immunology 101, Immunity 101 is why aren't you sick, right? And conceptually, our immune system is really divided into two functional arms, innate and adaptive. That's very different from other types of organ systems, which are actually organs, right? If I ask you what's the neurological system, you'll tell me it's the brain, it's the spinal cord. If you tell me, well, what's the respiratory system, well, it's the nose, it's your lungs. You can actually identify organs. But there isn't any organ of the immune system because they belong to other specialties, and that's a bit of perhaps a disadvantage when you're the patient, because in fact, sometimes you don't know which specialist you need to go to. But the immune system really is functionally an innate or adaptive function, right? The innate function means that these are based on preformed molecules. These are molecules that have what we call pattern recognition. So over the thousands of years of life on the planet, microbes have evolved parts of their molecule, what we call epitopes, that are highly conserved over time. And as a response, humans have their own molecules that recognize those microbial molecules, so that what you can get is once you see that bug, that pathogen, you get a response that's immediate, within seconds to hours, right? As opposed to the adaptive immune response, which is antigen-induced, and it's based on antigen recognition, and that response is delayed, days to weeks. So an example of an adaptive immune response is you get a vaccine today, and in about 2 weeks, if your immune system works well, you'll have antibodies to whatever was in that vaccine. That's different from your innate immune response, where if you go swimming in a swamp with a Komodo dragon, you don't really have time after you get bitten to wait days to weeks to have your immune response clean up that wound. You kind of got to respond now. And that's the innate immune response. Now, who does what? Well, in the innate immune response, we think things like complement pathways and neutrophils and monocytes and natural killer or NK cells are very important. They undoubtedly are. We'll get into that in a moment. But also, what's not circulating the blood is also important. The epithelium, your skin, your gut, your respiratory tract, those all see the germs of the environment before even some other parts of your immune system do. And so technically, they're actually part of your innate immune system. The endothelium are vessels that line your blood vessels because germs do try to sometimes get into your blood system. The components of your adaptive immune response is things that we classically have heard about: B cells, T cells, and antibodies. But at the end of the day, the mission is the same: sense and search, ignore if it's yourself, well, we're not supposed to be attacking ourselves, and destroy if it's foreign or not yourself. Now, that's the immune system. If the immune system doesn't work, that's called an immune deficiency, right? So it can be either inborn or acquired. An acquired immune deficiency is also called secondary immune deficiency, or SID. We call it secondary because we don't think it's inherently driven by genes, and that usually there's other factors that you're not born with that account for it. Aging, malnutrition, malignancies or cancers, certain medications that intentionally suppress the immune system, for example, in transplants, certain infections that we know can HIV, once it advances to AIDS, can lead to a secondary immune deficiency, or when you have other conditions where you lose components of your immune system. So, for example, certain gut diseases or certain kidney diseases, you can lose parts of your immune system. So therefore, the opposite of secondary would be primary. That's primary immune deficiency, or PID, or inborn errors of immunity, right? And that means that there is a gene that is responsible for the disease. Now, a primary immune deficiency, or PID, classically was recognized because it caused either fatal infections or life-threatening, not necessarily ultimately demised, but life-threatening infections. And since the 70, 80 years that they were first described, we now know that there's other things besides infections. There's something called atopy, which is predisposition to allergies. Immune dysregulation, so things that include autoimmunity, where the adaptive part of your immune system attacks yourself, or autoinflammatory, where the innate part of your immune system attacks itself, and then cell proliferation. Cells just can't stop dividing, and those are lymphoproliferative disorders and cancers. So that was the immunology part. Now, buckle up because it's going to get a little bit intense here with the genes, okay? But I think we need to understand it so that, we don't say the wrong things. These are single gene disorders. In other words, a single gene in your genome, in your 20, 25,000 genes that are in the human genome, are responsible for the immune system problem you have. It can be transmitted through what's called an autosomal recessive manner. Now, just to make sure we all understand that, that means that both copies of the gene are affected. So let me just take a step back. When you are born, when a human are born, most of the genes, there's 2 copies. 1 copy from mom, 1 copy from dad. And in a recessive disorder, both copies have to not function properly to have that recessive disorder. So when they don't function properly, we call that loss of function. That loss of function could be complete, like there's 0 function, or there's partial, anywhere higher than 0, but not, not anywhere near 100%. Sometimes you'll hear the word hypomorphic. Hypomorphic is just a fancy way for us to say partial loss of function. Autosomal dominant is not the same thing, right? A dominant disease is when among the 2 genes that you've inherited, only one of them is not functional, and that is sufficient for you to have the disease. Now, that impaired gene could have a loss of function that we just talked about, could be either complete or partial. On the other hand, that mutant gene, can also impair the function of the normal or wild type gene that we call dominant negative, or negative dominance, And sometimes you have what's called haploinsufficiency. In other words, when you have one mutant allele, you just don't get enough normal protein to have a normal function. It doesn't interfere with the normal protein. It doesn't have anything sinister, but just its absence makes you have insufficient amount. And so the insufficient, when you have to come up with a fancy scientific term, we call it haploinsufficiency, half of sufficient, and that's how you get the condition. But things have gone so funky over the last 20 years because in addition to a mutation impairing function, which is what most of us think, there's also mutations where you can have a gain of function. That molecule is doing more of it, what it normally does, but unusually more, that gain of function. So for example, a molecule can get phosphorylated, and if it gets too phosphorylated, that's a gain of function. That gain of function at the molecule level, however, is still bad because at the cell level, you still lose function. And then lastly, you have what's called new function or neomorphic. There's a mutation that confers onto a gene a new function that it's never done before. And then, of course, you can have X-linked recessive. Now, in addition to that, not everything is straightforward. Not everything in everybody's pedigree is exactly what the preceding or parental phenotype was. You can have what's called variable expressivity. And I'm using this example where a mutation in a gene can cause you to have the brown color, but as you can see, not all offspring have the same brown. Right? So that's what's called variable expressivity. Some have features of it, some have more features of it. And then you can have what's called variable penetrance. And this is a mutation in a gene that causes you to have an extra digit. So if you haven't noticed, the left hand has an extra thumb-like structure. But not everybody in that family who has that gene mutation will have an extra digit. So that's what we call variable penetrance. Within the same family, some people have express a phenotype or express a condition, whereas others don't. Right. That was the gene part. We're not even into the core of it yet. So now we're going to talk about microbiology. As a medical microbiologist, we spend a lot of time trying to diagnose infections, and the way we do that is very Cartesian. It isn't we take a sample of whatever, we put it into a machine and see what comes out, although sometimes it sounds like it. You have to have an approach. So what I teach my fellows and my residents is that approach. When we talk about germs, let's break it down into something scientific. First, we have viruses. Then we have bacteria. Then we have fungi. And then we have parasites. We're only going to talk about human microbiology. Trust me, there's a whole different world out there for animal microbiology, which I won't get into. Among the viruses, you can have those that are RNA-based. In other words, the genome of that virus is purely RNA. And I'll give you some examples. Flu or SARS-CoV-2, which is COVID. Right? Those viruses are RNA-based. That's different from DNA-based viruses, and the classic examples would be herpes virus, where herpes 1, things that cause cold sores in some people, herpes 2, VZV, which is varicella zoster virus, or the chickenpox shingles virus. Another class, separate from the herpes, is the HPV, which is human papilloma virus, which causes the warts. And the reason that's important is because from a clinical perspective, they cause different diseases. From a laboratory perspective, there's different ways to test it. Also, from a clinical perspective, there's different treatments. So what treats one won't necessarily treat the other. But I'll also explain to you that it's important from an immune deficiency perspective because it allows us to work it up further. Among the bacteria, we have gram-positive, things like strep and staph. And then you get more weirder bugs like nocardia and mycobacteria. You have gram-negative bugs like E. coli and Pseudomonas. Among the fungi, you have yeasts. Some people have heard of yeast infections. That's typically caused by Candida. You can have a special kind of yeast that infects your lungs if you have problems with T cells, which we'll get into, called pneumocystis or PCP. That's yeast, but you can also have molds. Mold is the fuzzy stuff that grows on your bread, right? And the classic example there would be Aspergillus. And then you have those that are both. They're what we call thermally dimorphic. So depending on the temperature, they could either be yeasts or molds, and those are actually found around the Great Lakes area and around a few other places in North America. And the classic example would be histoplasma. If you're from the southwest of the US, coccidioides may not be a foreign word to you. And then in parasite worlds, we also divide those up into protozoa, so things like Giardia and Cryptosporidium, and helminths, which are basically worms. Okay? Now, this classification is actually how we learn microbiology and infectious diseases. It's also how I think we should learn immune deficiencies. So let's go through some examples. If you take an RNA virus like flu or SARS-CoV-2, there are specific host defenses in the human body that answer to those types of viruses. What happens is that the virus gets in the cell, the RNA gets out into the cell, and it's recognized by several different innate immune pathways. We won't get into the details of the pathways or the genes or the molecules, but at the end of the day, those pathways then turn on that cell to produce something called interferon alpha. Interferon alpha acts on adjacent cells through a specific interferon alpha receptor. That receptor signals through its own molecules to tell the other cells, hey, the guy next door, he's infected by a virus. We got to do something. And what do we got to do? Well, first we got to put up our shield so that we don't get infected. The neighboring cells don't get infected. That's why it's called an interferon. It interferes with the viral infection. And we also got to kick butt. We got to go get that infected cell, and we got to go get rid of that virus so he doesn't linger around in our body. Now, if you have a mutation in any of the pathways that either can recognize the virus or respond to interferon alpha, you will likely have trouble with certain respiratory RNA viruses like flu or COVID. If you have a mutation in a molecule that affects not just this pathway, so what happens is, of course, if you won't be able to fight it off, but if you have a mutation in a molecule that affects not just this pathway, but it's a molecule that's shared with other immune pathways, well, then you can have susceptibility to this infection as well as to other infections, depending on what the other pathways are that are affected. That's RNA viruses in a nutshell. That's really all you need to know, frankly. I'll be honest with you. I tell that to my fellows as well. The weird thing that we found out is that with COVID, especially among elderly people, the reason there was a disproportionate amount of elderly people that had a catastrophic outcome to SARS-CoV-2 was that they had a defect in this interferon alpha pathway, but not a genetic defect. They had what we call autoantibodies. They produced rogue antibodies that are not thought to normally be there, and those autoantibodies targeted the interferon alpha and neutralized it and mopped it up, so that those elderly people were basically lame ducks, unable to respond when they got SARS-CoV-2 infection. And that's why there was a catastrophic outcome. A lot of this can be circumvented with antibodies generated by the vaccine, so that's good news. But this shows you that the convergence of studying genetic disorders can also allow you to study non-genetic immune deficiencies. Now, that was RNA viruses. What about DNA viruses like herpes or human papillomaviruses? They kind of start off the same, right? We know that there's this interferon alpha response, but they evoke a different, what we call effector arm, or arm that responds. Here they require more what we call natural killer or NK cells, as well as a special type of T cell called CD8 positive or cytotoxic T cells. And what those viruses do is that they recognize this virally infected cells, and they turn on this complex machinery on the left, which I won't go into, but on the right, it's basically the kiss of death, right? So those NK cells will go and find the virally infected cell. They'll connect to them. In a way, it's a bit of a kissing contact. It's a synapse. Bye-bye to that cell. That cell will then become killed. And that's the way you get rid of certain of the DNA viruses, because if you don't kill them well, they will often become latent. They'll stay in our body, fall asleep, and then they'll reactivate later when the immune system is a bit weaker. Bacteria. And we talk about bacteria, we can talk about a whole lot of infections, but I'm going to talk about the one that's most common to my clinic in the inborn errors of immunity is pneumonia. Pneumonia is an infection of the airways, particularly the lower airways of the lungs. Recurrent pneumonias is when you have more than one infection within a given time period. And sometimes the infection can spill out from the lungs to areas outside of the lungs, your bloodstream, or they can go to, example, your joints or the brain area. We call those invasive bacterial infections. Now, what happens is the following. Now, this is not drawn to scale, obviously, but this is a bacteria that enters into your airways, okay? And you have specific responses that are supposed to address that. Complement, neutrophils, and then your adaptive immune response. So we'll go through each of those. The complement pathway is probably the best way to torture medical residents, because what we say is, draw out for me the complement pathway. And this is what we expect from them. Now, the most obvious response you're going to get is this. But the reality is nobody really cares about what they draw. It's more just to make them sweat a little bit, But the reality is that there are distinct components of this pathway. And if you have a problem with the classical pathway or a problem with the alternative pathway, you can get things like pneumonias, recurrent pneumonias, and invasive bacterial infections. There's a very distinct part of this pathway down below, what's called the terminal membrane attack complex, or the terminal pathway, where for reasons that I don't believe are entirely still well understood now, several decades later, you can get an increased risk for Neisseria meningitis infections, or meningococcus. This is a bacteria that can cause meningitis. And in fact, I've actually had people, young adults, who present with such infections, and when you look for it, then in fact that's the reason why they have this infection. They have a terminal pathway defect. But the classical or alternative pathway, if you have a problem with either one of those, you cannot make one of those proteins that are in one of those two pathways, you can get pneumonias or recurrent bacterial infections. And the neutrophils are one of my favorite cells, I have to say. Probably because they have a finite lifespan of about 4 to 6 hours in the circulation. So, a short attention span works perfectly here. But these neutrophils, they develop in the bone marrow, and they go through specific designated stages as indicated here. So you can imagine that if you have a genetic mutation that decreases the production of neutrophils in the bone marrow, you will have low neutrophils, or if you really want to get fancy, neutropenia. That neutropenia can be sometimes called severe congenital neutropenia. It can be cyclic neutropenia. There's all kinds of neutropenias, and at the end of the day, you're just not making enough neutrophils. So the quantity is clearly abnormal, and that you could pick that up on a complete blood cell count blood test. You can see low neutrophils, there you go, and it has to be obviously a sustained event. But sometimes you can actually have quantitatively normal neutrophils. They just don't function properly, right? Sometimes I teach to a full room, but 3/4 of them are asleep. So just because you have a good attendance doesn't mean you have good performance, right? Same thing with neutrophils. I don't want to bring, students down to levels of, blood cells, but they sometimes are. You can have normal levels of neutrophils in the blood. They just don't function properly. So an example is something called leukocyte adhesion deficiency. Another one is called chronic granulomatous disease. Now, I wanted to go into some examples of how do you make the distinction between these. I don't want to gross anybody out, so just a small warning, okay? But if you have a neutropenia or if you suspect leukocyte adhesion deficiency, you can look for additional things. So you can have a baby who has infection around the belly button area, that's called the omphalitis, or the umbilical cord didn't properly come off at the right time. That's an additional clue. You can have a skin infection that doesn't heal properly, where you have basically a hole in your skin, a wound that doesn't heal. That can also be a sign. Or you can have things like perirectal, so this would be, if you can't make it out, it's like a bum. And around the bum area, you can have boils or skin infections or abscesses. Chronic granulomatous disease, CGD, is a little bit broader in terms of the types of infections you can get. You can get things where your lymph nodes, especially like in your neck, can become infected typically with a bacteria called Staph aureus. You can get that Staph aureus causing other infections in organs. So this would be a CT scan showing a liver abscess. And CGD also uniquely has the capacity or the susceptibility to an infection with the mold called aspergillus, where you get aspergillus of the lung airway and aspergillus pneumonia. So these are additional clues to look for in addition to the initial presentation I was telling you about. So that's it for neutrophils. What about adaptive immunity? Well, in the same way that neutrophils go through different stages of development, the B cells go through different stages of development. Part of it is in the bone marrow, where they go through these different stages. Part of it are in the circulation and in lymphoid organs. Excuse me. So if you have a problem in the development of T cells, a developmental defect, the best example would be X-linked gamma globulinemia, or XLA, you will not be able to produce B cells properly. There are a range of other B-cell disorders, particularly maturation defects, or we think that there are maturation defects. But at the end of the day, if you have a defect in anywhere along this pathway, you will not produce the end product, which is antibodies or immunoglobulins, and so you can have a range of problems. You can have A-gamma globulinemia, which is no immunoglobulins. You can have hypogamma globulinemia, which is low immunoglobulins. Then you can have low IgG2 or specific pneumococcal antibody deficiency and any other sort of permutation of antibody problems. Now, I make it sound like it's a B-cell issue, but it's not exclusively a B-cell issue. And I kind of use the analogy of like the T as in like for teenager, right? So you have a B-cell, and the B-cell thinks that they can do anything they want, and then at the end of the day, the B-cell can become the plasma cell and all that kind of stuff, and they think they're all tough and stuff like that. And you kind of have to remind the B-cell, but wait a minute, the T cells are around to help you. We're always there if you need anything. And do you need any money? They'd give you an eye roll, and any other offensive questions that you may ask them, right? So if you have a mutation that affects only the B cell developmental process, you will have the low antibody levels and immunoglobulins that we talked about. If you have a disorder that affects the T cell and its capacity to help B cells, you will not only have the low antibody levels, but you'll also have decreased T cell function. And if you have both decreased T cell function and decreased B cell function or decreased antibody levels, you have what's called combined immune deficiency. If it occurs at birth or shortly thereafter, that's a severe combined immune deficiency or SCID. If it occurs later, and I have a whole bunch of these people where it's recognized only later in adulthood, it's combined immune deficiency. I'm petitioning that we call it adult combined immune deficiency, but then the acronym becomes ACID and then, whatever. So we're gonna come up with some other fancy name. And so we actually discovered one that was due to a mutation in a gene called ICOS ligand. Not ICOS, that was well known before us, but now it's ICOS ligand, its partner molecule. So why is all this important? Well, the adaptive immune response produces antibodies, right? The antibodies in the airways to protect you from getting severe pneumonias. That's primarily IgG. There's some IgA. Now, some people say, wait, no, that's not what I heard. I heard IgA is really important at the mucosal level. And that's true up at the nose, right? So up at the nose, the upper airway, the IgA level is particularly important and out exceeds the IgG presence by a factor of 3 to 1. But in the lower airways, the ratio is a bit inversed. such that IgG is level at about 2.5 times that of IgA. So both are important, but IgG is much more present. Why is that important? Because it really emphasizes that if you're on immunoglobulin replacement therapy, where you're getting primarily, almost exclusively IgG, that's what we're doing, right? We're trying to pack those airways, the lower airways, the lungs, the alveolar space, with IgG so you don't get a severe pneumonia. And the corollary is if somebody has problems with pneumonias, including recurrent pneumonias, you should have been tested for things like complement, neutrophils, your B cells and your antibodies, and your T cell levels. Now, there are some funky germs that we are briefly going to get into, nocardia and mycobacteria. That could be seen with chronic granulomatous disease, as you also can develop Aspergillus pneumonia, which I mentioned. But this unique group of organisms can also present with a problem in the interferon gamma pathway, and they can also present with susceptibility to dimorphic fungi like histoplasma. So this is something that I worked on with in Steve Holland's lab at the NIH many, many years ago. But what we see is that if you have a monocyte or macrophage that gulps up the fungus, it produces IL-12, which then acts on T cells and NK cells to then produce interferon gamma and to close this pathway to try and kill this fungus. It also turns on a variety of other pathways, including adaptive immunity. Now, as the years have developed, over the 15 years or so since we started working on this, there have been mutations in these key molecules that predispose to impairing this pathway and therefore getting susceptibility to the nocardia, the mycobacteria, and the dimorphic fungi that we were talking about. We also ended up discovering a condition called, what at the time we called that mono-MAC, because the monocytes were missing and they were getting mycobacterial infection. That ended up now being GATA2 deficiency because it's due to mutation in GATA2. But you can also get the same infections if you have decreased production or function of T cells, because the T cells produce the interferon gamma. I'm not going to get into T cells because T cells are a scary story, and every month there's a new T cell, and I don't really understand it all, much less have the time to explain it. But just stay tuned. T cells are not a single entity. So at the end of the day, microbiology is very critical. We talk about, I'll just give you an example about a Candida infection, okay? These are people that I've actually studied in my clinic where this 30-year-old gentleman actually had seizures. He had a scan. He was told you have a cancer of the brain called glioblastoma. He continued to have seizures. He ultimately had surgical resection. This was apparently what they thought was going to be as good as it gets, and at 30-something years old, he was placed in palliative care. When you look at the biology of the biopsy, you're like, well, wait a minute, there's no cancer there. It was a fungus infection called Candida. So that's not normal, right? We all agree. If you can get Candida yeast infections, fine, but not in the brain. We did genetic studies. Long story short is we found a mutation in a gene called CARD9. We tested his cells and found that his cells cannot respond to Candida albicans by producing a certain hormone or cytokine of the immune system called GM-CSF. We therefore petitioned to give him GM-CSF, which exists to be used in completely unrelated conditions. So we gave him the GM-CSF treatment. We did serial lumbar punctures, and as you can see, the black and blue lines were abnormal on the left, and in the gray zone, they became all normal. This is his scan a few years later, and he continues to be alive to the point where the government caught on and said, wait a minute, you can't be palliative 8 years later, so they removed his palliative care privileges. And we've also found the same thing in other patients. This is another patient who had a very similar story, and this is a lady who actually had trouble with her vision, and long story short, ended up having Candida in her eye and in her brain. Lastly, just to finish off very quickly, we're going to talk about parasites. There's two that are very important. One of them is called Giardia, which some of you may have heard. We know that Giardia is problematic in people who have low, in fact, no IgA, so not a little bit low, but like absolutely no IgA levels in their blood. And that could be either because they cannot produce antibodies or they are just IgA alone cannot be produced. So if that's the case, we can take the paradigms that we just learned about and said, okay, well, if they can't produce any antibodies, they may have a B cell problem or a T cell problem, and they'll have not only low IgA, but they'll have low other antibodies. As opposed to if you look, you say, well, only the IgA is low and not the other ones, and that isolated IgA deficiency state, we don't quite understand the basis for that. Cryptosporidium is a problem, particularly here in the Midwest. You may have remembered that there was a huge outbreak of about 50,000 cases a few decades ago because of a break in the municipal water plant. But here, the problem with cryptosporidium is it causes persistent infections. It can even go to your biliary system, and the key cytokine for this is interferon gamma. So if you can't produce interferon gamma, then you can have really problematic infections with cryptosporidium. And that, as we talked about, can be due to defects in the IL-12 interferon gamma pathway, or you can't produce monocytes, or your T cells are defective. So in the last few minutes that we have, I just want to pose to you a quiz. None of you have answering pads. I'm just going to ask you to telepathically send me your answers based on the options that we have, okay? Just go ahead and do it very quickly. So what have we learned today? Option A, microbiology is painful. There's way too many bugs. Immunology is painful. There's too many cells. Genetics is painful. There's too many genes. Or D, we can use microbiology to understand where lies the weakness in immunology, and then genetics to understand why the immunology is weak, and that is really, really cool. So go ahead, just send me your answers, and we'll just tabulate it. I got just a few seconds. Oh, great, we have a 100% response rate. So let me just tell you what what I've understood from people. Some people think A, some people think B, some people think C, and overwhelmingly everybody thought D. So thank you very much. So just some key points as we finish off. Microbiology of the infections you may have is critical. Okay, one, it's important in the workup of your immune deficiency, especially if you have not been yet diagnosed. Do not accept the word infection. You push for the type of infection. You should push not only because it guides the investigation, but it guides your treatment. If you have a viral infection, you won't benefit from antifungal or antibiotic therapy, or vice versa. Right? So insist on microbiological testing of symptoms. Think of microbiology and immunology as a lock and key. And at the end of the day, when you have the intersection of these three specialties, genetics, immunology, and microbiology, you really get to the core of inborn errors of immunity. Thank you very much.
All right, so before we go to questions, just a couple of resources. The QR codes here go directly to them. So Ask IDF is our sort of online answering service. You can submit any question related to PI, and we will get you an answer or a resource or get you pointed in the right direction. If you're looking for peer support, we've got great Get Connected groups that are online. Some of them are based around diagnoses. Some of them are based around geography. So even though you're meeting online, you're meeting people in your area. Most important, please complete your session survey in the app. This is how we know whether you enjoyed this session or not. And that's how we plan for our educational content in the future. All right, so with that, I have my lovely colleague, Colleen, in the back, has the microphone. Please keep your questions relevant. Please do not share personal health information, and do not mention specific products, providers, or institutions. Thank you.
Okay, my question is about shingles, kind of the varicella virus. Can that show up later, like say you have it, and then show up later in life as kind of like a rash on a neck or a chest when you're like having an allergy flare-up or a common cold? And then also, could someone, like a parent, have CGD, but their offspring have CVID?
Okay, great. So two excellent questions, so I'll try to break them apart. The first one is the varicella zoster virus, or the chickenpox virus. And for those who didn't hear, the question is, can you have shingles or zoster come up under another situation where you're having some sort of stress? And the answer is yes. Now, we don't really actually know why there's shingles or zoster reactivation. We try to be all cute in medicine by saying, well, you know, your immune system's a little bit weak, and then the virus wakes up. But the reality is that it doesn't explain why the virus wakes up in a specific nerve root and then just goes along that nerve root, and it doesn't spread to other nerve roots, or why it doesn't wake up in other nerve roots. So I don't care what your physician will tell you, we don't know the answer entirely. But the context, however, is correct. We do know from epidemiological observations that there are different stressors that associate with reactivation in some people. So for some people, it's things like UV light exposure, so exposure to the sun, for example. For some people, it's actually a physiological stress. So they have a medical condition that requires hospitalization transiently, but clearly that must pose some sort of physiological stress on them that we can't really measure. And then shortly thereafter, they get an outbreak of shingles. So stress does permit reactivation of shingles, but why, we don't know. The second question is, can somebody have CGD and their offspring have CVID? And the answer is unfortunately yes. You're not, in a way, condemned to one inborn error of immunity. In the same way that, can somebody have Crohn's disease and their offspring have breast cancer? Yeah, unfortunately, that does happen, right? That being said, you have to also make sure that there isn't a misdiagnosis. Say, well, wait a minute. You're saying the person has a problem with their immune system, and you're calling it CVID, but did you know that I have CGD, and maybe what I have is what they have, and not well appreciated by the healthcare professional, and therefore may merit specific tests that wouldn't have otherwise been considered to make the correct diagnosis?
I am not a medical person at all, but
Neither am I.
I have.
No, I'm kidding. No. I did sleep at the holiday.
I have a reverse question. I know you guys do all kinds of wonderful work with all of our diseases, but my mother is 84, and she's never sick, ever. Have you ever done any research on healthy adults to find out what you can learn from them versus how they could help us that are sick? And why don't they get sick?
So, great question, for which the short answer is we don't know. But what we do know is a few things. We know that as much as these are genetic conditions that increase your susceptibility to infections, there's a whole other world that there are genetic variants that decrease your susceptibility or protect you from infections. So the best example would be the sickle cell trait that protects you against malaria. There's what we call genetic variants in a certain protein that coats not only your blood vessels, but your intestinal cells. And so from somebody's blood type, you can determine if they're at risk for certain types of norovirus infections, so that they're actually protected against norovirus. So that's why you'll see, for example, in some families, mom and offspring came down with a horrific gastro, but dad's sitting on the couch watching football and he's totally fine. It could very well be because of genetic bases for resistance as opposed to susceptibility. Why some people can live a long time and have absolutely no problems is something that we tried to specifically address with COVID. In the first few waves of COVID, as part of the COVID human genetics effort led by Jean-Laurent Casanova and Rockefeller and Helen Sue at the NIH, what we wanted to study were the centenarians, those who survived Spanish flu outbreak of 1918 to 20, who continue to survive up to this day, and has survived the SARS-CoV-2 pandemic. Not a lot of unfortunately, those individuals around, but that's what we wanted to study, because there is something to those individuals that, if you listen intently enough, there's a story that's being whispered. So that's what we're trying to study in general for resistance.
And that was the thing. My mom was around. Everybody in my house with COVID did not get COVID.
Well, if she's not vaccinated, we've got to sign her up for our study.
So quick question, when we're on IgG therapy and we still get sick, do you see a correlation between, environmental allergies playing into that at all?
Great question again. So a few things. If somebody gets sick, we have to kind of figure out what do they mean by that, right? Because sometimes people say things like, "I've got, a runny nose. I've got tickling in my throat." Sorry? Bronchitis and pneumonia. Well, even then, sometimes bronchitis and pneumonias can sometimes be asthma exacerbations rather than true infections. Or sometimes it's hard to make that difference. Sometimes it's things like, are their IgG levels even adequate for their condition, right? Because some people need higher IgG levels than others, depending on a variety of other factors. And so that's one of the things that, it really requires a tailoring, right? When somebody gets sick despite IgG replacement therapy, what are they getting sick with? The microbiology? Is it virus? Is it bacteria? Is it other things, non-infectious, like allergies? And then can we optimize, if it is an infection, can we optimize what they're on right now? Maybe their IgG level of 80 is not enough. Maybe they need a 10, 100 or something like that, right? So it depends a little bit on that person.
Okay, real quick, on your key points, you talked about the need to identify what the infection is rather than just, okay, here's an antibiotic. Can you give us any key points about how to advocate and what type of doctor would we go to? What do we tell them? Hey, I need to know exactly what it is.
Yeah, so, I get this question a lot because, people say, well, I was told there was a viral infection. Yeah, by who? By the Emerge doc did they do a test? No. Okay, so their definition of viral infection and my definition of viral infection are the same thing. I'm using the world standard microbiological definition of a virus, and they're using a treat 'em and street 'em definition of virus infection, which is get out of my emergency room, right? So what you need to do is you need to be an advocate. You need to say, I need to know if this is a virus infection. I can give you a sputum sample to see if it's a bacterial, and if they say, no, we're too busy, or it's not necessary, or whatever, advocate to see either an immunologist or an infectious disease specialist who will do those investigations. Chest X-ray, yeah. I mean, it's called microbiology for a reason. It's because you can't see it with your naked eye. I know physicians will tell me otherwise. That looks like a viral pneumonia. Garbage. That's just pneumonia. You need to do special tests. It's called micro for a reason. You can't see it. And so you need to do special tests to look under the microscope or in special molecular biology tests to look for the cause. I'm from Canada. I'm lucky I don't have to worry about that. I'm from Canada. Anybody who's sick, who needs investigations, gets it. I don't know what to tell you.
All right, thank you so much to Dr. Vin. We've reached the end of our time. If you still have questions, please submit them to Ask IDF and we'll get some answers to you. Thank you so much. Enjoy. The last session of the conference is across the hall.